After having discussed the sensory system and the motor system, now is the time to discuss the higher functions in the CNS. Let's talk about the hypothalamus. Very often the hypothalamic nuclei are asked in the multiple choice questions. For example, this actually hypothalamus is a very high yield topic. They can ask ventromedial nucleus is concerned with satiety, hunger, thirst, temperature regulation, or other way around, circadian rhythm is entrained in which hypothalamic nucleus? Suprachiasmatic nucleus, preoptic nucleus, anterior hypothalamic nucleus, posterior hypothalamic nucleus, something like that. So we got to understand and know by heart these nuclei and their functions and the effect of lesions. So let's make a table. The nucleus of hypothalamus, its connection or its fiber or its neurotransmitter associated with it, function of this nucleus and effect of lesion. Lesion, I know generally the lesions are hypersecretion or hyposecretion. We are going to discuss the hyposecretion, or rather loss of function type of lesion. Loss of function type of lesion we will discuss. So supraoptic nucleus. Supraoptic nucleus, these neurons have osmoreceptors in their walls. So there are osmoreceptors in the wall of these nerve cell bodies, and plasma, a hypertonic plasma, while it is flowing alongside these nerve cell bodies, water will come out because of the plasma is hypertonic. The cell will shrink, and that shrinkage of the nerve cell body will stimulate these osmoreceptors in the wall of this nerve cell. What's the function then? The function is synthesis of ADH, and then ADH will act on the collecting duct and will reabsorb water. What is the effect of lesion? For all these nuclei, we will see only the loss of function type of lesion. So here, loss of ADH will result in diabetes insipidus. To be precise, central diabetes insipidus or neurogenic diabetes insipidus. You know there are two types. Central, when there is loss of neurons that synthesize ADH, and nephrogenic diabetes insipidus, when ADH is present but its action on the renal tubules cannot be exerted because of the loss of receptor. So that would be nephrogenic diabetes insipidus. Here the lack of ADH due to the lesion will result in diuresis, and that's central diabetes insipidus. Second, paraventricular nucleus of hypothalamus. Paraventricular nucleus is involved in neuroendocrine reflex. For example, milk ejection reflex is a neuroendocrine reflex. Mind you, what is a neuroendocrine? What is this term? Neuroendocrine reflex would mean that the afferent limb of that reflex is neural and efferent limb is hormonal. That's a neuroendocrine reflex. For example, milk ejection reflex is a neuroendocrine reflex. So synthesis of oxytocin and the effect of lesion. Loss of oxytocin has certain effects. For example, one is delayed labor, postponed labor. Oxytocin, as you are aware, is involved in the process of parturition and labor. So that's the supraoptic and paraventricular nuclei. Many authors believe that there is no clear-cut distinction that the supraoptic synthesizes ADH and paraventricular synthesizes oxytocin. Many authors believe that both these nuclei synthesize both these hormones, ADH and oxytocin. Next, suprachiasmatic nucleus. Above the optic chiasma, suprachiasmatic nucleus. Now, there was an MCQ which was asking that if the visual cues are removed, what will be the effect on circadian rhythm? If there is no vision, visual cues are removed, what will be the effect on circadian rhythm of the body? And the options are: the cycle length will increase, it will become more than 24-25 hours. Cycle length will remain the same, normal, no change, or cycle length will decrease, less than 24 hours, or it will become extremely variable. The answer is cycle length will increase if the visual cues are removed, the cycle length of the circadian rhythm. Let's try to see what's this suprachiasmatic nucleus. Visual pathway, it gives one set of fiber to the suprachiasmatic nucleus of hypothalamus, and then this via superior cervical ganglion sends sympathetic fiber to the pineal gland. Pineal gland, which is called as epiphysis cerebri. If pituitary is hypophysis cerebri, then this is epiphysis cerebri. Also called as third eye. This is also, pineal gland is also called as third eye. So it is watching the external environment, the third eye, via this connection, and then its secretion is melatonin. So when we see the dark, when there is darkness, it is seen and it is conveyed to suprachiasmatic nucleus. Therefore, suprachiasmatic nucleus is also called as the link between external environment and body's biological clock. So that's the connection. And then when we see the dark, it is conveyed via the suprachiasmatic nucleus to the pineal gland. Eventually it will release melatonin. Melatonin then will circulate and will convey to all organs that it's night time. So metabolic rate will decrease and the functions will be altered. So that's how circadian rhythm is maintained and diurnal rhythm is expressed. So if the visual cues are removed, then can the circadian rhythm be maintained? Yes, it can still be maintained even if we are not able to see the darkness and the light, the night and the daytime. Still, how it can be maintained is by the skin temperature. I mean, that's one way possible, that we can feel the skin temperature, that in daytime it increases and night time it will decrease. The ambient temperature, I'm saying, will increase in the daytime, decrease in the night time. That will be felt by the skin, and those signals will be sent to the hypothalamus, and then the hypothalamus will convey that to the pineal gland. But the only thing is that this will take longer time compared to the visual input. This conveying via skin temperature will take a longer time, and therefore in the absence of any visual cues, the circadian rhythm will be there, but it will have a longer cycle length, more than 25 hours. So suprachiasmatic nucleus, what is its connection? Its connection is with the retina and visual pathway. Function is circadian rhythm. And what is the effect of lesion? Loss of circadian rhythm, for the want of better term, Jet lag is one term used when there is a disturbed circadian rhythm. Next, pre-optic nucleus. Basically, hypothalamic nuclei are on all the sides of this visual pathway. So supraoptic, preoptic, suprachiasmatic, and so on and so forth. Preoptic nucleus, anterior neurons are androgen sensitive and posterior neurons are estrogen sensitive. And so these neurons, the function is regulation of sexual function. Sexual function in males and females respectively by these anterior and posterior parts of the preoptic nucleus. What would be the effect of lesion? Effect of lesion is altered sexual functions. So loss of libido and altered sexual preferences. That's preoptic nucleus. Now next three nuclei can be remembered together. Make one club of those nuclei and remember them together. Let's see how. Ventromedial nucleus. Ventromedial nucleus. This is concerned with satiety. So, the neurotransmitter involved in the satiety is CART. We have discussed this at the start of the discussion on CNS, that CART is a neurotransmitter which is known to play the central role in satiety. Cocaine and amphetamines regulated transcript. It's a neuropeptide. So ventromedial nucleus is concerned with satiety. What is the effect of lesion? No satiety. So there will be hyperphagia and obesity. Hyperphagia resulting in obesity. That's ventromedial nucleus. So just remember like this: if the ventromedial is concerned with satiety, then lateral hypothalamic area is concerned with hunger. Lateral hypothalamic area. These neurons are said to be orexigenic neurons. Orexigenic neurons. Anorexia is loss of appetite, orexia is hunger. So orexigenic neurons are the hunger producing neurons. Well, these neurons are also said to be glucostatic neurons. Glucostatic neurons, they can sense the blood glucose, and as the blood glucose falls below a critical level, these neurons begin to fire, and that is how one starts feeling hungry. So these neurons are concerned with hunger. What is the effect of lesion? Loss of appetite. Anorexia, loss of appetite, loss of weight. That would be the effect of lesion. And the third nucleus, if remember like this, lateral hypothalamic area concerned with hunger, then lateral superior hypothalamus is concerned with thirst. Lateral superior hypothalamic area or nucleus. These neurons have osmoreceptors again, just like ADH synthesizing neurons, osmoreceptors in their walls, and when there is a hypertonic plasma which circulating alongside these neurons, these nerve cell bodies. Then water will come out, the cells will shrink, and that's how the neurons will trigger the thirst. So thirst. Now, absence of thirst, again, for a want of better term, it's called as adipsia. If excessive thirst is called as polydipsia, then this would be termed adipsia. So adipsia, that's a thirst regulating nucleus in the hypothalamus. So these are important hypothalamic nuclei. One or two are still left. For instance, periventricular nuclei. Now, periventricular nuclei, there is some argument regarding these nuclei as to whether they are involved in reward and punishment type of behavior, or anger and rage reaction. A few authors believe that this is concerned with reward and punishment type of a behavior. So that's one. And second is rage reaction. This is concerned with this nuclei are connected with the limbic system, and they are connected with the limbic system, and they are involved in reward and punishment and rage reaction. Rage reaction of an animal. What will be the effect of lesion? No rage whatsoever, so timid animal. But mind you, there is certain amount of debate whether this nucleus is involved in the rage or it is involved in the reward and punishment type of behavior. And finally, last one nucleus in the temperature regulation. So most anterior and most posterior hypothalamus. Anterior hypothalamus and posterior hypothalamus. Anterior hypothalamic area, the neurons are warm sensitive, and posterior hypothalamus, the neurons are cold sensitive. So the stimulation of these neurons, the anterior hypothalamic neurons, stimulation of those neurons will result in cutaneous vasodilation and sweating. Cutaneous vasodilation and sweating. That is, when the body temperature had increased, these neurons will trigger this response and the temperature will be decreased. Cold sensitive neurons, when they are stimulated, when the body temperature is falling, then posterior hypothalamic neurons will be stimulated and they will trigger shivering to increase the body temperature. So the function, if you see the function, temperature regulation, the function of these neurons, temperature regulation, and the effect of lesion in the anterior hypothalamus, the lesion will result in hyperthermia because the body temperature could not be decreased after the lesion, and lesion of the posterior hypothalamus will result in hypothermia, as the shivering response is not possible, increasing body temperature not possible, it will result in hypothermia. These are the hypothalamic nuclei. So basically, hypothalamus is the part of the brain. It is a part of diencephalon, which has two parts, the thalamus and the hypothalamus. Thalamus, we have already discussed that it is a sensory relay station for all the sensations. So there are those thalamic nuclei. We have discussed those relevant nuclei in different parts. And the hypothalamus is concerned with vegetative and visceral functions of the body. We have seen these nuclei and their functions. Apart from that, hypothalamus is the highest seat for ANS regulation. the highest seat for ANS control is in the hypothalamus. So these are the hypothalamic nuclei and their functions. Let us also see some of the other higher functions. For instance, the hemispheres and their functions, learning and memory, and then sleep, EEG, and the other functions. So, hemispheres. There are two cerebral hemispheres, as we are all aware, dominant and non-dominant. These hemispheres, this is a common knowledge now, that in right-handed individuals, left cerebral hemisphere is dominant, and in left-handed individuals, right is dominant, or both are equally dominant. Right and left hemispheres equally dominant in left-handed individuals. Now, because this terminology was not so very accurate, therefore the new terminology came in for these hemispheres. So dominant hemisphere is now called as categorical hemisphere, and non-dominant hemisphere is called as representational hemisphere. Representational hemisphere. So that's a new nomenclature. Now, categorical hemisphere is concerned with language function. It's concerned with speech. I mean, Broca's area, that's the motor area for speech, is present in the dominant hemisphere. So it's concerned with speech, language, etc. Whereas non-dominant hemisphere is concerned with representation of body parts, orientation of body parts with each other, spatio-temporal correlation of body parts with each other. And therefore, we have already seen this in the sensory system, that lesion in this hemisphere, in the parietal lobe, non-dominant hemisphere, if it is lesioned, then it results in hemi-neglect syndrome, because orientation of body parts with each other is lost, and with the immediate surrounding is lost. Therefore, it results in hemi-neglect syndrome. That's when a person neglects the opposite half of the body or opposite immediate surrounding. If the person is wearing some shirt, only one side of the shirt will be worn, and the person forgets to wear the opposite half, something like that, or ignores the immediate surrounding. There is one area called as planum temporal. Planum temporal is an area in the temporal lobe, and it is seen in the dominant hemisphere. It is larger in dominant as compared to non-dominant hemisphere, and this asymmetry, that it is larger in one hemisphere compared to other, this asymmetry is said to be exaggerated in musicians. Musicians who have a perfect pitch, they have a perfect understanding of the pitch of the songs. They are in them it was observed that the planum temporal in the near the auditory cortex, in the temporal lobe area 41 and 42, they have a very large planum temporal area in the dominant hemisphere compared to non-dominant hemisphere. So these are some of the nomenclatures in the hemispheres. Let us try to understand the learning and memory. What are the various types of learning and memory? Another higher function associated with the cerebral cortex. Well, let's start with learning. Learning is acquisition of new information and skills is called as learning, and storage of that information is called as memory. So acquisition of a new information, new skill, the learning, and storage of that skill is the memory. Right. Learning is of two types: non-associative learning and associative learning. Non-associative learning means, first let's see associative learning, that the person associates two or more events and learns. That's the associative learning. For example, we see the dark clouds and then it rains, it pours and it wets us. So we learn this, associate these events, and then next time when we see the dark clouds, we carry an umbrella. That's how we learned. So associative learning. So we have associated two or more events and learned, whereas non-associative learning means single events. We learn from single events. Let me just make one point before I tell you these categories. All the types of learning and memory are basically the presynaptic modulations. There is going to be modulation of presynaptic neuron in the learning and most forms of memory, except long-term memory. Long-term memory that's formed in the hippocampus. In that, there is modulation of the postsynaptic neuron. So just remember like this: most of the types of learning and memory involve modification of the presynaptic neuron and its behavior, except for long-term memory, in which there is modification of the postsynaptic membrane, postsynaptic neuron. So let's see these types. Non-associative learning, there are some examples like priming, habituation, sensitization. These are the types of non-associative learning. I'll just give you one examples for these. For example, what is habituation? Decreased response to a stimulus after its repeated exposure. The response decreases if the stimulus is unimportant. That's called as habituation. The person learns to ignore that stimulus because it is not important, therefore it will be simply ignored. The person learns to ignore it. For example, if you are going in a new place, initially there is ticking of the clock and the person is unable to sleep because of that ticking sound. But over a period of time, the person learns to ignore. So that's called as habituation. And what is the basis for habituation? Decreased release of excitatory neurotransmitter by the presynaptic neuron. There is decreased release of neurotransmitter by the presynaptic neuron. So transmission at the synapse is decreased, and that's how person will learn to ignore that event. What's sensitization? Sensitization is opposite actually. Is a stimulus which is said to be life-threatening or very important, the response is increased or enhanced actually, because the stimulus is either very important or life-threatening. So sensitization, how does it occur? Increased release of neurotransmitter by the presynaptic terminal for a particular stimulus. That's sensitization. Over a period of time, the neurotransmitter release increases, and that's how the response to a particular stimulus is increased. Example, a breastfeeding mother, while she is fast asleep during the night time, No other sound will make her get up from the sleep, but cry of the baby, and then the mother will immediately get up due to that sound. So mother is sensitized to a particular stimulus. That for that particular stimulus, the neurotransmitter release is increased over a period of time. That's the sensitization. Right, these were non-associative types of learning. Associative type of learning means the person learns from two or more events. Associating two or more events and learns. Basically, there are two types in this. One is classical conditioning by Pavlov. Classical conditioning by Pavlov. And operant conditioning. Operant conditioning by Skinner. So, what's classical conditioning? We know the experiment already. A dog experiment, when dog is presented with the food pellets, there is reflex salivation. This is an innate response. This is an innate reflex. When the dog is given food, there is reflex salivation. This is an innate reflex. But now we see how the dog learns. Therefore, what we do is there is ringing of the bell, and then the food is given. You see, there is an unconditioned stimulus and there is a conditioned stimulus. Unconditioned stimulus is giving of the food. This is a natural stimulus. And conditioned stimulus means to which the dog is going to be trained. The dog is going to learn that stimulus. That's conditioned stimulus. So that is ringing of the bell. And how the pairing is done. There is pairing done. Conditioned stimulus, ringing of the bell, immediately followed by the unconditioned stimulus. So these two are paired like this. Conditioned stimulus immediately followed by the unconditioned stimulus. Eventually, over a period of time, the dog will learn, and then just for the conditioned stimulus, like that ringing of the bell, there will be reflex salivation in the dog. So that's how the dog learned that when the bell will be rung, that means food is going to be presented, and therefore only on ringing of the bell there is reflex salivation. So these are the terminologies used. Conditioned stimulus should be immediately followed by unconditioned stimulus, and this pairing should be done repeatedly. There are a few things that should be noted, that if after a certain period, if this training is stopped, or if there is a lot of interference between conditioned and unconditioned stimulus, if there are other stimuli, or there is a lot of time gap between conditioned and unconditioned stimulus, then there would be extinction of the training. After a while, this training may be extinct and it will disappear. So this is the classical conditioning. Now, what is the difference between classical conditioning and operant conditioning? Try to understand this. In classical conditioning, there is reflex salivation. That is, it's a reflex response. The dog does not perform anything actively. Whereas in the operant conditioning, the animal actively operates on the environment and learns. Understand the difference between the two. In classical conditioning, there is only reflex response from the animal, whereas in operant conditioning, animal is going to actively operate on the environment, and that is how the animal is going to learn. So a monkey kept in a cage, commonly known as the Skinner's cage, going in one compartment, there are electric shocks which are delivered, and going into the other compartment, the bar hits head of that animal, but then immediately food is presented. So the animal, while operating on the environment, learns that in one compartment there is punishment and the other compartment there is reward. So after a while there is a positive reinforcement for the reward and negative reinforcement for the punishment. So animal has learned that where it should go and where it should avoid going, negative reinforcement and positive reinforcement. That's operant conditioning. These are the basic types of learning. Let us also discuss the memory. There are three types: short term, intermediate term and long term. Short term and intermediate term have almost a similar type of physiology, so we will discuss short term and then the long term. Short term memory has two physiological bases. One is post-tetanic potentiation. Post-tetanic potentiation. If I ask you to remember something for a while, or 2 or 3 phone numbers, 10-digit numbers, what would you do? You will go through the numbers very repeatedly and rapidly, very fast, rapidly. You will go through the numbers for 5-6 times, and then for some time you can recall those numbers. Post-tetanic potentiation would be the basis. If you stimulate a synapse tetanically at high frequency, when you are actually rapidly going through the numbers 4-5 times, you are stimulating a synapse tetanically. Tetanically means at high frequency. So what happens is, with each stimulus, with each impulse, calcium kept coming in, and therefore the calcium accumulates for some time. And therefore after that, means post-tetanic potentiation, after that, whenever the impulse arrives in this synapse, there will be extra release of neurotransmitter, because you know the amount of neurotransmitter released in a synapse is proportional to the amount of calcium present in the presynaptic terminal. So extra neurotransmitter is released, and therefore extra transmitter release means the number is recalled which you were by hearting. Or one more basis given for short-term memory is presynaptic facilitation. Presynaptic facilitation. We have seen presynaptic inhibition, that's seen in the axonal synapses. Similarly, there is presynaptic facilitation as well. Here is a synapse. It's an excitatory synapse, axodendritic synapse. As we all are aware, axodendritic synapse is where facilitatory transmission occurs. So this synapse is meant for transmission of impulse. Now imagine one more axon comes and ends on this axon. It releases the excitatory transmitter, so much so that there are extra calcium channels opened and extra neurotransmitter released at this main synapse. This is called as presynaptic facilitation. Just as we saw presynaptic inhibition, that one axon comes and it decreases the release of neurotransmitter at the main synapse. Similarly, there can be presynaptic facilitation. So this is a temporary phenomenon, and therefore it is the basis for short-term memory. But you can notice one point, whatever events that we discussed so far in the case of learning or memory. For example, when we discussed habituation and sensitization, habituation was because of decreased neurotransmitter release by a repeated stimulus which is deemed to be non-important, and sensitization was increased neurotransmitter released for a stimulus which is important. Or over here, all these events are presynaptic events except long-term memory. Long-term memory is basically a post-synaptic modification of behavior in the postsynaptic neuron. So, long-term memory. The basis for long-term memory is long-term potentiation in hippocampus. Long-term potentiation in hippocampus. We remember there are structural changes in the long-term memory. There are no structural changes in the other types of learning and memory, but there would be structural changes in the development of long-term memory. There will be changes in the dendritic geometry. New synapse has been formed and there will be a change in dendritic geometry. There would be increased neurotransmitter vesicles. There would be increased release sites for those neurotransmitter vesicles. So all these are the structural changes, and therefore it needs an increased amount of protein, increased protein synthesis. So what happens in the long-term potentiation in hippocampus? Well, basically it is glutamate, the neurotransmitter, and it is acting via its NMDA receptor in the case of long-term memory. So how it is developed? You see, first of all, cortex develops a working memory. Cortex will first develop a working memory, some short to intermediate term working type of memory, and then these neurons project on to the hippocampus. Particularly a few areas noted in the hippocampus, such as CA1 region of hippocampus. So what will happen is, this is a postsynaptic neuron. There would be long-term potentiation in the hippocampal neurons. That is, the excitation of the hippocampal neurons will be seen on a long-term basis. The neurons will have extra impulse propagation. So that's hippocampus, where long-term memory is formed. Long-term memory is formed. Rather, working memory is converted into long-term memory in the hippocampus by increased synaptic transmission in the hippocampal neurons on a long-term basis. And as I said, glutamate transmitter and NMDA receptor on the postsynaptic membrane are the associates. Once this is formed, then it is consolidated in the neocortex. So if you are asked, long-term memory is formed in the hippocampus, but it is consolidated in the neocortex. Long-term memory gets consolidated back in the cortex, neocortex. That's the long-term memory. What happens is a long-term potentiation, as I said, glutamate, as it is released, it combines with its receptor, and there is sustained opening of channels, cation channels in the postsynaptic membrane, and there is a sustained influx of calcium in the postsynaptic membrane. Remember, excitatory transmitter glutamate via one of its receptors increases the calcium influx. So sustained influx of calcium, and therefore EPSPs, excitatory postsynaptic potentials, are developed on a long-term basis. Sustained development of EPSPs in the hippocampal neurons in the postsynaptic membrane. So as I said, long-term memory is changes occur in the postsynaptic membrane. One last point is, what is the effect of lesion of hippocampus? Effect of lesion of hippocampus will result in Anterograde amnesia. You see, amnesia is of two types. Retrograde amnesia means the person will completely forget what has happened up to that point. The past memory will be wiped out. That's retrograde amnesia. Anterograde amnesia means, which happens after the lesion of hippocampus, it means a person will not be able to form the long-term memory from that point onward. So, no new memory and no new long-term memory will be formed after that point, from that point onward. So that's the anterograde amnesia. Long-term memory cannot be formed by the hippocampus. This happens in the hippocampal lesions. So that's about the learning and memory. These are some of the higher functions. Let's also see the sleep and EEG. They always ask questions on the EEGs. They will give you EEG record. This is very common. They will give you the EEG record and ask you to diagnose the EEG record. For instance, what is this EEG record? You should be able to read those waves and then accordingly diagnose what has been given. These are 2 alpha and 2 beta waves. The options given are, is this a record of wakefulness or REM sleep or stage 2 of the NREM sleep or stage 4 of the NREM sleep? This is the record of wakefulness, 2 alpha and 2 beta waves shown. Let's try to understand some EEG waves. Well, there was one question recently asked. Highest frequency waves on the EEG are which waves? Alpha, beta, gamma, delta. Well, the answer is gamma. 4 waves which are already known are alpha, beta, theta and delta. So what is this gamma wave then? Gamma oscillations were discovered only a few years ago. High frequency oscillations of the baseline. So that was the answer. Let's try to understand the EEG. EEG record is the record of cortical potentials, record of the potentials in the cortex, recorded from the surface by putting surface electrodes. That's electroencephalogram. And one more point, these are synaptic potentials. Remember, EEG record is a synaptic potential. It is not an action potential. EEG records do not show action potentials. These are synaptic potentials. They are synapses in the cortex which are sending those potentials which we record. So these are EPSPs and IPSPs, and these are not the action potentials. Right. What are the waves in the EEG? Let us discuss the waves in the EEG. Alpha wave, 8 to 12 hertz, 8 to 13 hertz. So you will see 8 to 13 waves in a cycle. That's the alpha wave. 8 to 13 cycles in one group. These are the waves of quiet wakefulness. Quiet wakefulness with eyes closed and mind wandering. Please try to understand that the beta waves are the waves of, see, alpha is the wave of quiet wakefulness. Beta waves will be the waves of alert wakefulness with eyes open and mind focused sharply. So these waves are the waves of quiet wakefulness with eyes closed and mind wandering. Mind is not focused, mind is not fixed on something. This is also the stage when you go to bed and try to fall asleep. There is a latency when the eyes are closed but you are awake and mind is wandering. That's when this alpha wave can get recorded, or when you're seated quietly during daytime hours. So even then, these waves are recorded from occipitoparital region. Occipitoparital region. It is thought that when the person closes the eyes, there are temporary after images of the things that he saw just before closure of the eyes, and those after images may be responsible for these waves, so mainly getting recorded from the occipital lobe. That's alpha waves. Then when the person opens the eyes, beta waves will be recorded. So 15 to 30 hertz beta waves are low amplitude and high frequency waves. 15 to 30 cycles will be seen in one group. That's a beta wave. They are the waves of alert wakefulness with eyes open and mind focused, mind sharply focused. So these waves, they are recorded from parieto-frontal region. Parieto-frontal region. Now, listen to this. Beta waves are also recorded in REM sleep. For which reason? For this reason, REM sleep is also called as the paradoxical sleep. REM sleep, the rapid eye movement sleep, is full of paradoxes. You know, the person is sleeping. This is one paradox. Person is sleeping, and yet the waves of alert wakefulness get recorded from his cortex. That's a paradox, and REM sleep is full of such paradoxes. So it's called as paradoxical sleep. And therefore, remember this. For the sake of MCQ, answering, if they give you all the beta waves on the record, then the answer should be REM sleep, because REM sleep records beta waves. If there are all alpha waves, 8 to 12 hertz, then either this is the stage 1 of the REM sleep, that is when the person is gone to bed and trying to fall asleep, he will go through the successive stages of NREM sleep, stage 1, stage 2, stage 3, stage 4, and then there is REM sleep. So this is the starting point of the sleep during the latency period, when the person has closed the eyes but is awake. That time alpha waves. So all 4 alpha waves can be said to be stage 1 of the NREM sleep, or start of the sleep, when the person has actually not fallen asleep. All 4 beta waves, then the answer should be REM sleep. And 2 alpha and 2 beta waves that's the EEG of the wakefulness. 2 alpha followed by 2 beta waves. So the point is, mainly there is alpha rhythm, and when it is disrupted and beta waves come in, they replace alpha waves. This is called as EEG desynchronization. It's a misnomer, yet it's called EEG desynchronization. And this is the EEG for wakefulness. This is the EEG of wakefulness. This is how you can see how many waves are there, and accordingly you can conclude that this is alpha wave or beta wave, and then come to the conclusion of whether wakefulness or REM sleep or NREM sleep. Okay, theta waves. Theta waves, 4 to 7 hertz. They are the waves recorded in NREM sleep, non-rapid eye movement sleep, also called as slow wave sleep. These waves are recorded in children from the temporoparietal region of especially children, because children's cortices are less active, less developed as compared to adults, so therefore they have slower frequency waves, the theta waves, 4 to 7 hertz. So it should appear like this. Slower frequency and slightly more amplitude, so 5-6 waves in one group. That should be the theta waves. 5-6 waves per group, that's a theta wave. Theta waves are also recorded from hippocampus, and they are recorded in the stage 3 of the NREM sleep. Stage 1 and stage 3 would show the theta waves, and then delta waves. Delta waves 0 to 4 hertz. Now what is the meaning of this 0 hertz? What I'm trying to say here is, even if the thalamocortical projections are cut, still these waves will be recorded. So even if there is no arousal coming from the neurons which are ascending, you see the thalamus sending the signals to the cortex, and there is ascending reticular activating system. These are responsible for the arousing and alerting responses. They cause arousal of the cortex, and that's responsible for these waves, various waves from the cortex. So the point that I'm making is, from the thalamus to cortex, if these connections are cut, still some waves will be recorded from the cortex, and they are delta waves. So very slow frequency waves. And remember, high frequency waves will get recorded when the thalamocortical projections are very, very active, and ascending reticular activating system is causing arousal and alerting response for the cortex. Then it will be alpha and beta waves. Delta waves, very slow frequency, when even when no signals are coming from the thalamus to the cortex. These waves, okay, 0 to 4 hertz, so just 1 or 2 in one group. These waves are recorded in organic brain disease. Remember, EEG is recorded to diagnose the functional brain diseases, like epilepsy is a functional brain disease. But when there is some organic lesion, there is some space occupying lesion, or some tumor or such disease, some lesion, infarction, etc., then in organic brain disease you get the delta waves. Remember, otherwise EEG is generally meant or recorded to diagnose the functional diseases of the brain, like epilepsy, as I said. These waves are also recorded, the delta waves, in deep sleep, deep coma, deeper planes of anesthesia. So, when the cortical activity will be bare minimum, almost nil, that time these slowest frequency waves will be recorded. And therefore, they will be recorded in the stage 4 of the NREM sleep, which is the deepest part of the sleep, stage 4 of the NREM sleep. So, these are the various EEG waves. Let's first see gamma oscillations. Gamma oscillations, 30 to 50 hertz, Gamma oscillations are high frequency oscillations. They have been discovered recently in last about 10 years or so. The gamma oscillations, high frequency oscillations of the baseline, and they are recorded when a person is having a very high mind activity, solving some very complex puzzle or complex arithmetic, and a very high mind activity focused on something, you will get gamma oscillations. That's the highest frequency waves. Let us see the sleep and various types of sleep and sleep pattern, etc. You see, what is a normal sleep architecture? The normal sleep architecture is like this. It's a cycle of 120 minutes. One cycle is of 120 minutes, and such 4 to 5 cycles will occur in a night's time. So how do you see these cycles occurring? And what is a cycle composed of? You see, first, when a person goes to bed and trying to fall asleep, there would be about 90 minutes cycle of NREM sleep, also called as slow wave sleep, non-rapid eye movement or slow wave sleep. And then there would be about 15 to 20 minutes of one episode of the REM sleep. And mind you, it happens like this. These 90 minutes will be that stage 1, stage 2, stage 3, stage 4 of the slow wave sleep. The person is going into deeper and deeper parts of the sleep. And then it becomes reverse. This pattern will be reversed, means from deep to superficial it will come, and then it will enter into the REM sleep. So that's a normal sleep architecture. When a person goes to bed trying to fall asleep, he will go through the stages of NREM sleep 1, 2, 3, 4, in which the person is going deeper into deeper and deeper sleep. Then it is reversed, the order is reversed, the sleep pattern, it becomes superficial, and then it enters the REM sleep. So that's one cycle, and then this cycle will be repeated 4-5 times. That's how that's the normal sleep architecture. Well, when the person goes into sleep, there would be first the NREM sleep or slow wave sleep, as I mentioned, non-rapid eye movement or slow wave sleep, because slower waves will be recorded in this sleep. 4 stages. Stage 1. Now I am assuming that a person has just gone to bed and trying to fall asleep. He is awake but trying to fall asleep. Mind is wandering. Then it would be alpha waves. Or if it's in the later part of the sleep, then it would be theta waves. So that's the stage 1. Then stage 2. Stage 2 of the NREM sleep. That has a very classical type of EEG. It has been repeatedly asked in various entrance exams. There is sleep spindle followed by large K complexes. Sleep spindle followed by large K complexes. That's a pattern, that's the EEG in the stage 2 of the NREM sleep. Then stage 3 and stage 4, that is the person going into deeper and deeper sleep, would be the delta waves. Deepest part of the sleep will have delta waves. The deep sleep, as we've already discussed about it. After this, it will go in the reverse order. The sleep will become superficial and then enter the REM sleep. That's how the sleep pattern occurs. So let's see some of the concomitants of the REM sleep. This MCQ has been asked a number of times. Which of the following is not a concomitant of the REM sleep? which of the following is not a concomitant of the REM sleep, means it occurs in the NREM sleep. And the options given are penile tumescence, nightmares, somnambulism, and heart rate and respiration irregular. Somnambulism will not occur in the REM sleepwalking, because somnambulism will require muscle tone. REM sleep, there is loss of muscle tone. Let us discuss this. REM sleep, what are the concomitants? Rapid eye movement, what other things are seen? Well, as I said, it is also called as paradoxical sleep. It is full of paradoxes. One of them I already mentioned, beta waves get recorded on the EEG. Beta waves are the waves of alert wakefulness, but they get recorded in REM sleep also. Then what else? Heart rate and respiration becomes irregular. We must have experienced this once in a while, that when we get up from the sleep, heart was pounding or respiration was very rapid or irregular. That's the REM sleep. Then loss of muscle tone, sleep paralysis sometimes called, loss of muscle tone throughout the body. We also often experience this, that we felt completely helpless during some part of the sleep. We wanted to scream at a, let's say, thief, but just voice could not come out, or we wanted to get up, but it was completely helpless situation. That was because REM sleep, when loss of muscle tone. There is loss of muscle tone throughout the body, but you know which muscle group loses the tone maximally? It's that muscle group which does not get rest and relaxation during the waking hours. So which muscle group do you think would be this? It is the muscles of the neck extensors. You know, all our muscle groups, other skeletal muscle groups, they get the rest at least partially during the daytime hours. But neck extensors never get the rest, because our head is always upright, and therefore they lose the tone maximally. There is a maximal loss of muscle tone in the neck extensors. You will experience this if you are traveling at times and the person accompanying you is sleeping. You will see that very often the neck extensors are losing the tone and the head is dropping intermittently once in a while. So that's greatest loss of tone. And no loss of tone occurs in which muscle groups there will be no loss of tone, means they maintain their tone. There are at least 3 important muscle groups. One is diaphragm. At least respiration will be continued, so diaphragm maintains the muscle tone, its tone. Second is extraocular muscles of the eyeball. Therefore there are rapid eye movements, for which reason it's called as REM sleep. And middle ear muscles. So you see, this happens very often, that you are sleeping and you are in the midst of a dream and somebody calls in reality, and that gets mixed up with your dream and some mix-up happens. So because your middle ear muscles were intact, their tone was there intact, and therefore the sound did reach your cortex and it got mixed up in the dream. And then tongue muscles, they wouldn't allow the tongue to fall back. So these are the muscles, main muscles that do not show the loss of tone. Otherwise there is loss of tone throughout the body. Apart from this, you will see dreaming memory consolidation. Memory consolidation of the dream and memory consolidation of the previous days events. Remember, it very often it will happen to you that you were into a dream throughout the night, but you just couldn't recollect it in the morning. You can't recollect it in the morning, although you saw the dream throughout the night or most part of the night. Why you can't recollect in the morning? Because major part of it has happened in the REM sleep and there was no memory consolidation which could happen, so you can't recollect, because memory consolidation happens in the REM sleep. REM sleep therefore is very good so far as the memory consolidation is to be concerned, for the long-term memory. Nightmares, very bad dreams. Nightmares occur in this sleep, whereas night terrors will occur in the NREM sleep. Penile tumescence and penile erection also occurs in the REM sleep. In fact, this is used by the clinical psychologists to differentiate between organic impotence and psychological impotence. If penile erection is there in the person in the REM sleep, then the impotence is just psychological, and if it is not there, then probably it is organic impotence. So that's these are the concomitants of the REM sleep. NREM sleep, there is no loss of muscle tone in the NREM sleep, so you see such features as night terrors. The person literally thrashes about, kicking and boxing and all that, so that's night terrors. Somnambulism, sleepwalking, somniloquy, sleep talking, somniloquy, sleep talking. So all such events which require muscle tone are seen in the NREM sleep or slow wave sleep. Bruxism, there is a fair amount of debate as to whether it occurs in the REM sleep or NREM sleep, because bruxism, the teeth grinding, it needs a certain amount of muscle tone, and since we are saying muscle tone is lost in the REM sleep, it must be occurring in the NREM sleep, the bruxism or tooth grinding. However, many authors suggest that bruxism occurs in the REM sleep, the tooth grinding. So we may go with that, bruxism in the REM, and all these occur in the NREM sleep. So that's another important segment in the higher function discussion, the sleep and EEG. Some of the higher functions of the brain include emotions or emotional basis of behavior. So to understand that, let's first understand the division of the cortex. 90% of our cortex is neocortex, and that neocortex has all these cortices which we have discussed in the CNS, frontal lobe as motor cortex, parietal lobe, occipital lobe, temporal lobe. So that all is the 90% of the cortex. Well, and that part is called as neocortex, whereas the remaining 10% is called as archicortex. So obviously this goes by the evolution, newer in evolution, and archicortex is older in evolution. That's why neocortex and this the archicortex. So this 10% part includes the limbic system. Limbic system is the part which controls our emotions. At the center of it is the hypothalamus. So many of the emotions like rage reaction, reward and punishment type of behavior, these are discretely controlled from some nuclei or connected with some nuclei of the hypothalamus. And around this there are about 10 to 11 structures, small collections of nuclei around this. those structures will be collectively called as the limbic system. So hypothalamus and the structures surrounding it, small structures, this makes the limbic system, and this forms the basis of our emotions. So the structures like cingulate gyrus, hippocampus, mammillary body, amygdala. Amygdala is said to be the emotional window through which we see the external world. So all these parts have been given some small functions in the emotional context. Then one important facet of it was the Papez circuit. James Papez was the neuroanatomist, and in early part of the last century he put forth this Papez circuit, and this was said to be this was put forth to give the emotional basis of our behavioral responses. Why do we behave in a certain manner, and what is the emotional basis to it? That was given by this Papez circuit. So it gives the connections between the neocortex and the archicortex or the limbic system, and therefore the circuit thus goes like this. Our neocortex, or to be precise, the prefrontal cortex, is connected with the cingulate gyrus. From here on, the circuit involves the structures of the limbic system. So the neocortex or prefrontal cortex connected with the cingulate gyrus, which is a part of the limbic system, which is then connected with the hippocampus. Hippocampus, as we've already discussed in the discussion videos, is the part where working memory is converted into long-term memory. So all our past emotions will also have to be stored in the form of long-term memory. Therefore, hippocampus comes into the circuit, which is then connected to the mammillary bodies. Mammillary bodies, as in hypothalamus, the part of hypothalamus. Now, you know, as I said already, hypothalamus has a central role to play in some of the emotional aspects, the rage reaction, the reward behavior and all that. So hypothalamus is taken into the circuit. It also has the other vegetative functions along with the emotional functions, so that is integrated into the circuit. Then there is mammillothalamic tract. This is the mammillothalamic tract, which will then take the signals to the thalamus, anterior thalamic nuclei, because thalamus is the sensory relay station for all our sensations. They have to pass through the thalamus to reach the cortex, and therefore all the somatic sensations which are integrated in the thalamus, that also needs to be taken into this circuit. After all, behavioral basis of emotions will include the sensory inputs from the past and from the present. So that's where thalamus comes into the circuit, and then the circuit is completed by thalamus sending back signals to the cingulate gyrus. This is a to and fro connection with the neocortex and limbic system. So that's the Papez circuit, and these are the structures involved and the basis why these structures were taken into the circuit, because they have the certain role to play. Like hippocampus will have the long-term memory of our past emotions, and so on and so forth. So that's the Papez circuit, and it explains the emotional basis of our behavioral responses.